We work to create a world
that's free of infectious diseases.
We work to make sure
that no infectious disease
is left unchallenged.
We discover, develop,
implement and evaluate
health solutions,
working alongside the most at-risk communities.
Doing so ultimately builds
a healthier, safer world for everyone.
We are the Kirby Institute.
Hi, everybody. Welcome. Thanks for joining us for one more seminar under the Kirby seminar series. And this is a global health themed seminar. We are past 1 o'clock, so I guess I cannot see how many participants we have online. Do you mind? Is it okay for me to start? Yeah, okay. Okay, so I mean, you know me, but maybe people online don't. I think this is an open seminar. My name is Susana Vajneri. I lead the Neglected Tropical Disease Research Group at the Kirby Institute in the Global Health Program. And I'd like to begin by acknowledging the traditional custodians of the lands on which we gather today and pay my respects to their elders past and present. We are presenting from Bedegal people of the Eora Nation. And I extend that respect to Aboriginal and Torres Strait Islander peoples who may be joining us today here as well in the audience and online. I think most of you will be familiar with the format of these seminars, just some housekeeping notes again. So it's an hybrid presentation. We are here, but we also have people joining online. So Jake will present his seminar, and there will be a Q&A session at the end. For those of you here, just raise your hands and you're free to talk and ask your question. Otherwise, please, for the people joining online, please use the Q&A button at the bottom of your screen, and you can type your question and I can read them out loud at the end of the presentation. So it's really my great pleasure to introduce Jake Baum, a professor of infectious diseases and head of School of the Biomedical Sciences at UNSW. Jake and I go back around 10 years when our paths crossed when I was in London. He had recently moved to Melbourne and we shared... 20. Oh, 10, 20, sorry. It is, okay. Yeah, I know, I look quite young still, but yes, 20 years ago, when we were both doing malaria research and had interest in the same sort of proteins expressed by the malaria parasite that I'm sure Jake will present on. But anyway, it's really great to have you here and be, with having this opportunity again to collaborate more closely in the future. So Jake is a parasitologist, a cell biologist, and innovator in infectious diseases translational research. He did a bachelor's and master's degrees at Oxford, and then a PhD at the London School of Hygiene, and then he joined the Walter and Eliza Hall Institute of Medical Research in Melbourne to work on the molecular and cellular biology of the malaria parasite. And that's when he co-discovered PFRH5, those proteins that I was also interested in. And this protein is actually the current lead of field blood stage malaria vaccine. And that's a discovery that facilitated establishing his own independent lab in 2010. So once again, I'm very curious to hear about this very interesting topic and be back in the end for questions.
Thank you very much. Right, I share my screen. Hopefully that's coming through clear. I know it is clear here. Is it clear online? All good online. So thank you very much. And it's a pleasure to be joining you from the Kirby. And I believe that the phrase is Bujari Gamarwa, which is Gadigal language for welcome or good day. And I'm trying to do my bit about connecting to local lands. Today, I'm going to give you sort of a bit of a scattergun introduction to some of the work that's been going on in my lab, which we're going to be continuing here. Hopefully, should I click? So I'm sure many of you with an interest in global health will be very familiar with malaria and its kind of annual statistics. We just had the revamp from last year's World Malaria Report. I think the main take-home theme there is that the statistics for malaria haven't improved. They're now kind of in this sort of flat line. The number of infections each year, the number of deaths each year is sort of quite stable, which is not a good thing. And so it still has an incredible impact. There are various, sources of extra concern on the horizon. There's the spread of artemisinin resistance, which has now been reported in Africa. So there's kind of the spread of resistance to our frontline radical therapy. It's not complete resistance, but it's still worrying in terms of the ability to treat the sick child. So that's a study that came out now over nearly 2 years ago. So I actually don't know what the latest is, but clearly drug resistance for the parasite is a major concern for us. There's also vector resistance to insecticide. That's a major concern. I think it's also worth thinking about COVID. I often describe malaria as, in the old analogy, a canary in the mine. The idea of the thing that tells you that society is falling apart in a tropical country is often the rise in malaria cases. We've seen that politically around the world. And COVID really showed that too, that, best estimates are that in the midst of the peak of the pandemic, hopefully we're past that, but the midst of the peak of the pandemic, an estimated extra sort of 13 to 14 million extra cases, 60,000 deaths arose from malaria because of that impact on COVID, on health infrastructure as a result of COVID. So malaria is often a really good litmus test of how well we're doing in global health in general, as well as being a cause of concern itself. I really like this study that came out of the Imperial College Modeling Group, which started to sort of explore this question of, if we keep doing what we're doing, what will happen to malaria over time? And they predicted this stagnation. And I think the real take-home message from this and from other studies is that if we really want to do this thing called bending the curve, or reducing malaria incidence, but down to levels where we can say we're on the road to control, elimination, eradication, those kind of words, we need to innovate. We need to do things that we're not doing currently. So we need to come up with new strategies, new drugs, new diagnostics, new cures, new ways of intervening with the parasite and its life cycle. And that's this sort of big red arrow saying, if we want to get down, we're going to have to innovate. And that's now become quite a constant call from the leadership, both from the WHO, but from also the malaria leadership in the world, that if we really want to end malaria for good, we've got to innovate. So my lab's traditionally from a sort of a foundation of cell biology has been really interested in just trying to branch into innovative areas, or to try and be innovative, to move into new areas that may have implications in both fundamental understanding of the parasite, diagnostics, drug discovery, and also vaccinology. And so I want to tell you 3 stories about that today. I just thought I'd briefly start with a bit of life cycle biology. You can't give malaria talk without a life cycle, but also I really like this. This is an opportunity to really just show you some of the beautiful cell biology that malaria parasites have as they go through their life cycle. So if we start off with blood stage biology, we're dealing with a eukaryotic parasite. So it's a cell which is as complicated as one of our own cells, which invades a red blood cell. And this is a movie that one of my students took of a merozoite, it's about a micron in size, sidles up to a blood cell and enters. This is one cell invading another cell, but this is a eukaryotic cell invading another. I liken it to pushing an orange into a balloon. It's quite a magic feat to be able to do that. Later in the life cycle, when the parasite's been in a mosquito stomach, it then goes through rapid sexual maturation. And this is the process where the male gametes are formed. So this is like formation of sperm, happens in 20 minutes. It's one of the fastest, if not the fastest mitosis known in eukaryotes, produces 8 flagellated male sperm. They then fuse with the female. They produce this fantastic motile zygote called an ookinete, which is Bob Sinden in London describes as a tank that literally pushes its way through the mosquito's stomach and forms a cyst there. They then burst, rupture, and release these other forms called sporozoites, which are then injected during a mosquito bite. This is my favorite movie from Rogerio Mino, which is a proboscis being filmed going into a mouse ear, and it's something from Psycho, squirting out saliva to stop a blood clot whilst releasing sporozoites in green into the dermis of the animal. So you can, and then they then go to the liver, invade the liver, go through another round of sort of metamorphosis, and then start to invade red blood cells again. and this is another part of Rogeri's work, where you show, where you can see a sporozoite, which is in the dermis, crawling around the architecture, finds a capillary, circles the capillary, breaks through the capillary, and then goes off to the liver itself. It's a really, really terrific cell biology throughout its whole life cycle. And I just like to put up those 4 images of a merozoite, a gamete, an ookinete, and a sporozoite. This is the same parasite. This is just, I guess this is just epigenetics. This is the same genome being expressed in different ways with different cells, but beautiful cell biology. So if you ever wanted to, inspire a student to do a project in cell biology, there's a lot of fodder there in the malaria life cycle. Okay, so let's tell three stories. The first is a story about thinking about how we might find new drugs for malaria, and then I'll get on to diagnostics and vaccines. So when you're thinking about malaria control, we often think about the sick child, the child that turns up in a clinic, and this is data well over 100 years that correlates the fever pattern of a child, the temperature they're going through, with the types of parasites that you'll find in their bloodstream, the asexual forms and the crescent-shaped forms, and those are the sexual stage forms. So what's clear is that for most drugs that we use in clinic, a child goes into clinic, they're sick, they're treated, they end up being a happy child, and they go home. The thing is about most antimalarial drugs is they kill the asexual forms, but they don't kill the sexual forms. So that child then goes home, but is fully competent for transmission of malaria in their community. So if you only focus on cure, you do great things for the child, but you don't do anything necessarily for community transmission. And as we've all become kind of well rehearsed, transmission is often the secret to breaking the back of an infectious disease. So if we really want to think about transmission, we need transmission blocking drugs. So I inherited a group in London when Bob Sinden retired, which included Michael Delves, who's in the top right, and together with a series of team, but in particular Eliza, who has really helped us sort of really develop the high throughput version of the screen. We were screening for drugs that inhibited male and female gamete maturation, mimicking the conditions of the mosquito's stomach, fooling the parasite into thinking it was transmitting, and then trying to block the ability to then fuse and form the next stage of the life cycle. The idea being is if you could take a drug, that drug would be in your circulation so that when the mosquito fed, it would be transmission incompetent. It wouldn't transmit the parasite. And because of the skill of these researchers, we were able to get this up to about 14,000 compounds per week in a screen in a flat well plate, filming exflagellation, which is very active, and also filming female rounding up, which is a singular moment where they release this red protein on the surface. So we screened thousands of compounds, and we get these beautiful dose response curves from different drugs, some of which are male specific, some of which are female specific. So artemisinin are actually very good transmission blocking drugs. They target males, but not females. Some drugs, like the ones down the bottom, you can see they have activity against both males and females. So we did a Gates-sponsored drug screen with GSK and Dundee University using the Dundee University Global Health Diversity Chemistry Library, something like that, GHCDL, Chemical Diversity Library. And we screened up to about 70,000 compounds. And the idea was to try and triage and find as many different new chemotypes that could be the backbone of future drugs which were transmission blocking, either as drugs on their own right or as drugs that we might then combine with asexual drugs to treat. And so here are some of them that we then put into feeding assays with mosquitoes to show that they inhibited transmission of the parasite to the mosquito. And we even took some of them into mouse models of malaria, where we could show, if you just look at the far right, you can see that some of these drugs, I should just put the laser pointer on. You can see that some of these drugs, if you give them to a mouse 1 hour before feeding, they actually block transmission. So kind of proof of concept that you can give a mammal a drug and then feed that mammal about an hour later, hopefully longer when you develop the drug, and actually be able to block transmission. So that was quite exciting, in particular for some of the new classes of drugs that we discovered. One of the things, again, being a cell biologist, I really like to look. This is what happens in normal male gamete development. So you start off with a male gametocyte, which is elongated, falciform in Falciparum malaria, which is where Falciparum malaria gets its name from. So crescent-shaped. It goes through this rapid double mitosis and then forms axonemes, so the flagellum, and then they burst out. And then what was nice is that when you add different drugs give different phenotypes. So they work in different ways. They mess with different aspects of the cell biology of the parasite. And one in particular caught our eye because we found 6 different members of the same family. It's a sulfonamide backbone and has this R group, which can be modified quite a bit, but the drug still retains its activity. So we've decided to focus quite a bit on that drug. And I'm not going to talk about the extensive work we did on this 881 drug backbone, but actually it just came out today in Disease Models and Mechanisms. So if you want to have a look at the work. And what Sabrina, a PhD student in the lab, did is she worked tirelessly to use click chemistry to find the target of this drug and show its mechanism of action. And so we found that PFS16, which is a well-known marker of gametocide development, looks like a very solid target for this drug. So we now have a mechanism, we now have a target, so development should be a little bit easier. I think there's still, for me, and a question I want to keep asking my group, and I keep asking myself is, so what? You know, you found a great drug, looks like it's not very available, bioactive, it kind of gets degraded after about an hour. Is this ever really going to be a drug? If we're going to really walk the walk of innovation, we've got to be, you know, tough on ourselves and say, did we just discover a drug which is just a paper, or did we actually discover a drug? So we gave it a lot of thought. We're not med chemists, we did work with med chemists, but how would we turn this into a real drug? How would we actually deliver that transmission blocking drug? And what was really nice is Sabrina did a short sort of mini sabbatical over in Flaminia Cataruccia's lab in Harvard. And they're really pioneering this idea of using drugs in either insect baits or in bed nets to ask the question of, could you have an anti-malarial in your bed net that goes through the body of the mosquito and actually like a prophylactic drug for the mosquito to stop it from getting infected. And what was really nice is 881 works actually really well in these conditions, that if a mosquito is exposed to this drug, it actually will absorb it and will block transmission. That's sort of unpublished work, but that's kind of work that we hope will come out relatively soon. So in the future, one could imagine that, yes, you might be able to have a drug that you could take, which would block transmission, but if we can't get it sort of into the human, maybe we can get it into the bed net or into a sugar bait, into some kind of platform where we'd actually block transmission. So that's an area which I think is I'm quite excited by, because I think that it's much easier to develop a drug for human use if you don't have to go through humans, much less sort of safety barriers. Okay, that's the new drug discussion. I could take questions, but I think I'll go through to the end and then feel free to ask about the different sections. Okay, diagnostics. I have to say, I was never really interested in diagnostics, but that really changed with COVID. Suddenly everybody, I think we all have to pinch ourselves that if you know, there is virtually, there's almost not a house in Australia, probably not a house in most countries in the world that doesn't have a molecular diagnostic test in the kitchen cupboard. I think that's remarkable. Anyway, okay, so diagnostics. I think when you're thinking about diagnostics in global health, for diseases which are in rural or low to middle income countries, you really have to think hard about what you're dealing with. You're dealing with a lack of resources, a lack of infrastructure to actually, undertake the diagnostic, the process of diagnosis, a dispersed healthcare system. So bringing diagnosis and treatment together is not easy. Often the quality of tests might be quite low. Integrating that idea of diagnosis and treatment, which was already mentioned, with care provision. And in the context of a disease like malaria, you can't afford to wait. You can't have a child that is febrile, it's not well, and say, could be sepsis, could be malaria, could be something else. Let's send a sample to the lab. You can't do that. So most of the time, a clinician will just treat, or you've got that moment of indecision. So I think thinking about that, and this is a paper that a group of us wrote for PLOS Digital Health, which is really exploring this idea of what does the future of diagnostics need to look like for developing world diseases like malaria. I think another thing I'd just like to put out there in the diagnostic space, which really blows my mind, is this idea of HRP2 resistance in malaria. And this is where parasites have evolved loss of a gene in response to the selection pressure of being diagnosed. I think that's mind-blowing, right? So that's parasites out there from being diagnosed by a rapid flow test have stopped producing the antigen that the rapid flow test is based on. So that's a bit like COVID suddenly losing the spike protein, because that's the protein that we use in the rapid flow. The parasite is still there. It's evolved. I think that's really crazy, and it just really shows you that you can't be complacent even with something as simple as diagnosis. So we've been rethinking diagnosis for a while, and in particular, I wanted to just talk briefly about this collaboration with the group of Pantelis Rodrigo at Pantelis Giorgio at Imperial College, who's the second face along here, and his team, Kenny, who's the PhD student who led the work, with Jesus, our long-term collaborator, Aubrey, and Nick, all part of this Lacewing collaboration. And this is a platform based on LAMP, so not PCR, so it's a non-cycling sort of relative of PCR. And what the idea was is to use LAMP technology to amplify a parasite molecular signature, DNA, and interface that with a silicon chip based on the chemistry that Pantelis and his group have really pioneered. And couple that as a diagnostic with a smartphone, and then you'd get this kind of rapid, portable, molecular test for something as complicated as malaria. And this is the chip that they've developed, and it interfaces really beautifully with a mobile phone app that Nick has designed and their team, and that's kind of the whole lacewing platform. And the beauty of it is that DNA amplification is linked to a chemistry change, basically proton release, I believe, and then that interacts with the silicon chip, and then that tells you, and then that then communicates and tells you sort of diagnosis information. So what we did is we designed 6 independent LAMP assays for the different potential parasite species. So we wanted to get species level identification on chip, and I'll go through this pretty quickly because it's been published. But we were able to get as good a results as we were getting with RT-PCR in terms of specificity of amplification and detection. What was really nice is you can tweak primers for particular drug targets, and you can make them SNP dependent. So you can actually say, I'm going to amplify the Kelch13 gene, which is one of the markers of artemisinin resistance, and I'm going to have LAMP primers that differentiate between different mutations, which tell me whether this is artemisinin resistance or sensitive. And you can do the same thing. So now this is bringing in not just diagnosis, but potentially drug resistance diagnosis onto this same platform. And then the whole reason we did this was not because we wanted to have a clever molecular test, but because it was this point of care idea. And so we took this platform to Ghana. We didn't do it in a clinic, but we did it in a lab next to clinical samples, and basically had 6 devices where we were adding DNA extracted samples. That's now hopefully moved towards blood samples being on blood samples on chip, where you add them to the device, you get the change in pH relating to amplification, and then that then sends the message to the mobile phone. So you've literally got 2 devices which are about the size of a mobile phone, and that's telling you whether somebody's got malaria or not, potentially whether they've got drug-resistant malaria as well. And so that's even the old version. The new version is white and looks even a bit more like an iPhone. But the idea is to get the cost of the cartridge down, cost of the assay down, and you're talking about something, it's still probably too expensive at the moment, but something like $20 a cartridge that could not only, and because of the chip, you just divide the chip up and you could get multiple diagnoses in one chip. So you could have a febrile chip, which says the child has a fever because A, B, C, D, E, F, G, whatever. Or it could be, we know it's malaria, we just want to know what kind. And this could be also applied to any myriad of other diseases. And actually, we just got a small grant to try and do this for dermatophytosis, a fungal infection in Australia, together with Atian Tatian from the Sydney Children's Health Network. And the idea is to really just start to play with this idea of, could we start to develop molecular diagnosis for myriad diseases? And this is work that Pantelis' group is doing for things like dengue, COVID, and other diseases. So that was kind of the techie side of things. Whilst we were getting techie, we got really excited because we had the best call I've ever had in terms of funding, which is where a friend of mine who had connections with the Gates Foundation just phoned me up one day and said, "How much data do you have?" I was like, "I don't know, loads." What kind of data? He goes, "I don't know, I'll call you back." And I get another call a day later saying, "But how much data do you have? Do you have like a lot of data?" I go, "Well, I've probably got, quite a bit of data." But what do you mean, like imaging, sequencing? He goes, "Imaging, let's say imaging data." And the conversation just went on. And eventually what it turned out is that the Gates Foundation were keen on pairing research groups with Google to see if we could use machine learning to drive something in the malaria agenda. So we ended up pitching this idea of screening for drugs using high content imaging and AI, and it was really cool. And it's a paper that we're really proud of. It's probably not going to go anywhere because it's just too complicated. It's like really, really clever. So we had all these ideas of what was going to happen next, but then of course, COVID hit. So this work was led by Mira Davidson and George Ashdown, who've now moved on from the lab. COVID then hit and we were stuck. But it actually gave us an opportunity, something which we hadn't planned on doing, which I think is much more impactful. And that's to use the technology that we were developing, but for something as rudimentary as a Gimsa smear. So if you haven't got a rapid diagnostic test in Sub-Saharan Africa, in India, in areas of the world where you want to diagnose malaria, you actually use a technology which is well over 100 years old by Herr Professor Gimsa, who invented a stain which you put on red blood cells. And if they're infected with malaria, they go purple. They get this wonderful purple. You can see maybe you can't, these lovely purple rings inside red blood cells. So this is a really old technology. There's Gimsa. It's really old technology, which is still used around the world with just a standard microscope. So could we use this AI approach to identify infected red blood cells and then use that as a diagnosis against malaria. So again, I'm not going to go through this laboriously because it has been published. You can feel free to look at it. But we effectively came up with a strategy which would try to improve what people had tried to do before. So we had a 3-model approach. Can we find a cell? Can we find an infected cell? And can we tell what stage of the parasite life cycle it's at? So it's sort of a 3-stage model built around machine learning. And it was COVID, so we didn't have a lab, but the rest of the world, some labs, in particular in Australia, were open. And so collaborators in Adelaide and in Queensland, in Thailand, in the US, in Germany, sent us as many images as they could of Gimsa smears. And one of the beautiful things about the project, and we were already primed because of the collaboration with Google, is we wanted messy, varied, different data. We didn't want the same exact conditions for each smear. So we got really heterogeneous data, and that was really important in terms of building robust models. And then we went through these rounds, this 3-model: object detection, infection, and stage differentiation. What it really showed me is how bad humans are in terms of consistency when you're trying to diagnose something. So humans are really inaccurate. So here is our singular image, just one of many that we were sent, of parasites inside red blood cells. So we ask our humans, labeler 1, how many of these are parasites? So labeler 1, pretty confident, not quite sure about this one in blue. So this might be a parasite, might not be a parasite. Labeler 2, missed this one entirely, but had the same uncertainty about that parasite there. Labeler 3, clearly somebody with a lot of uncertainty, maybe a nervous disposition, doesn't like to make a call. So this, they only called one parasite, the rest they were uncertain. Compared with labeler 4, extremely cocky, knows exactly what a parasite is. You know, I know what a parasite is, 5 out of 5, right? And labeler 5, who must have got distracted, maybe mid scoring, was asked whether they wanted coffee and they looked away and missed 2 parasites. Anyway, so humans are extremely heterogeneous. The model, and it's not that the model is necessarily right or wrong, but the model is consistent. The model will always call those 5 cells infected versus uninfected. And so you can kind of start to see that when you look at all the data. We used to have these cupcake parties where people would just label relentlessly cells to build our ground truth data against which we could sort of develop the models. And then the really nice thing is when you ask the model to stage things, and this was really, it's quite hard to communicate why this is so beautiful, but this is a computer that's saying, this parasite looks a bit like this one, and this one looks a bit like this one, but this one looks more like this one than it looks like this one. So therefore I'm going to put them in an order. And that's all it did. And what it does is it recapitulates the asexual blood stage life cycle in terms of order from early to late, just by virtue of it getting more and more purple. And other features. So the 3-stage model can detect and do that. And then for me, the really, again, so what? Great paper. Is it useful? So Mira built this application called Plasma Count, which is a website where you can upload your data. It's actually a movie of this happening. So you can take, say, 10 images on your iPhone, and you can do it on your phone, down the camera, down the lens of the microscope. You then upload your 10 images. The machine then whirls. You go and have a cup of tea. You get an email saying your results are ready right now. Any minute, there you go, there's your email. And then you come back, and then you've got your data gets spat back at you. And it's not just that the data is there and it's quantifiable, but it's that you can explore the data. So if you were a clinician and you just had a slide scanned, and you can actually go through and make sure that you agree that's a parasite, not a bit of fluff. So I think there is some real utility there that you could then interface this with a microscope. And again, those microscopes already exist in the developing world. It's not, we wouldn't involve any new technology, and most people now do have access to a smartphone or something with a camera that could be used in that context. So the idea would be to standardize Gimsa counting, not only for lab use, but potentially deployable for field-based diagnosis. And I really like this idea of that it archives the data. I think there's a real problem in smear collection is that smear is a glass slide that is either kept or dropped. And there's no real archiving, there's no real ability to go back and consult with an expert whether that really was a parasite or not. So yeah, smartphone, down the lens, potential diagnostic for malaria for use in the field. I just want to put out one caveat here, is the machine, the website has recently crashed, the student has left, and I don't have any funding to continue. I really want to. So if there's anybody listening who's got loads of money and wants to continue this work, I'd love to keep it going, but we do need computer science students who understand how to code and know what GitHub means and things like that, which I don't. Just the PI, I don't understand how it works. But anyway, yeah, little pitch there. Okay, last but not least, vaccine. So if you've been watching the malaria, if you've been watching the malaria story, last year was a big year. So after like 40, 50 years of pushing this agenda of malaria vaccine, finally RTSS ASO, the vaccine from GSK, Mosquitix, was licensed for global use. Its efficacy, if you compare it to what we're kind of used to language-wise in terms of COVID, it's 30–40%, and it wanes very rapidly. So after about a year or maybe even 6 months, immunity wanes, and it's going to need that constant booster. I mean, in some ways, COVID is in the same situation. That's not going to work for malaria in the long term, but at least it's going to have some short-term massive impact. 30–40% efficacy when you're dealing with 600,000 children a year dying is pretty huge. There are plans afoot to redevelop RTSS. One of them is this VLP called R21, which is effectively the same thing, but just more parasite antigen. It looks exciting in terms of efficacy. There's a bit of debate about the conditions this was done under. But even here, you still got this idea of waning immunity, that immunity to a malaria vaccine, that this current malaria vaccine is waning. So I'm not an immunologist, and I'm really scared about saying these kind of things in public, especially in front of esteemed immunologists, because I'm likely to get it wrong. But I'm going to just go out there on a limb and say what I'm going to say anyway, and you can attack me later. But why does efficacy wane? Well, in my thinking, it's just you have to have this army of antibodies in peripheral circulation that are ready, and you need enough of them to stop parasites getting through and then going to the liver and then causing infection. And if I just remind you of that video with parasites being squirted out into the dermis, it just takes one of these parasites to get through, and you're going to get liver stage infection, which then leads to the exponential growth of blood stage infection. So this is the stage against which this vaccine works. You're going to need that high titer of serum antibodies in order to stay protected. And if that titer wanes, you're not protected, and there's no time to produce new antibodies. You just need that army to be present. So that's my understanding of why efficacy wanes so quickly. So if antibodies clearly only have a short time to act, you need very high titers, and if one parasite slips through. And I just like this image that Nina, a master's student in the lab, took. This is of a salivary gland which is full of sporozoites about to bite, just to really drive home that message that there could be an army onslaught of sporozoites, and you need to be really ready to receive them if you're going to go with that antibody route. The other thing is the target of the vaccine is kind of, I sort of wonder whether the parasite's been playing this great trick on us. Now, the parasites had millennia to evolve immune evasion strategies, and it's put 1 million copies of this protein called CSP on its surface. It's evolved out throughout mammalian evolution. In fact, even chicken malaria parasites have CSP on their surface. So the whole of vertebrate evolution has been a game of evolving this protein. It's got a lot of repeats in it. And if I were a parasite, I would evolve a protein that was so seductive that not only would the immune system go after it, but vaccine makers would go after it and spend their entire careers chasing that singular protein on the surface. So maybe there are other proteins we should be thinking of, and we're not really thinking of, because it's just so easy to go after the spike protein of COVID, right? It's sort of the analogy, the biggest, juiciest protein on the surface. And I know that's also hampered other diseases where they've tried to make vaccine against the obvious candidate, and sometimes the obvious candidate has got strategies to avoid. So another strategy that I think is really important to consider in vaccinology for malaria is the role of T-cell immunity, in particular in the liver, and some great work that's come out of the Melbourne group, in particular the work of Daniel Reed-Fernandez from Bill Heath's group, on the role of tissue-resident memory CD8 positive T-cells in conferring protection from malaria. And if you can get enough T-cells resident in the liver that are protective, then you have that chance. You've got a 6-day window to stop malaria parasites maturing in the liver and then go on to blood stage. If you combine that with then the early deterrent of an antibody, I think you'd have a much better vaccine. So antibodies have that short window to act, but cellular immunity has days to act in the liver. And if that's probably how the best vaccine we have works. And the best vaccine we have is to immunize people with the whole parasite itself. So if you take irradiated sporozoites or sporozoites under chemical to a drug coverage, you can actually get sterile immunity against malaria. It's challenging, and there's a company in America that's actually marketing a whole parasite vaccine scenario, but it's not an easy thing to do. And one of the key challenges of a whole parasite vaccine is that it requires dissectors to look down a microscope to dissect salivary glands and to pull out the sporozoites from inside those mosquitoes. That's a real challenge. Scenario have done it, but it's still a massive challenge to produce the material. Could we isolate the parasites without dissection and make a manufacturable vaccine? So that was a question that Josh, up in the top right, a really smart postdoc in the lab, asked. Could we develop a dissection independent method of sporozoite isolation? to really make a manufacturable whole parasite vaccine. And so he effectively came up with this method where you take a tube full of mosquitoes, you put them in a blender, you come up with a mosquito smoothie, you then pass that mosquito smoothie through some filters, you put them through this liquid chromatography, which is a technology developed in the 1960s for bovine artificial insemination, or used for that. And what's really cool is it takes one person 120 minutes to 2 hours to do this whole process cascade, but it doesn't involve any dissection. And it's literally just making a smoothie and then passing it through these filtration steps. And what was really nice, again, this is published, so you can have a look at this in your own time. What was really nice is that the sporozoites we were getting out of it, as they go through this 4 or 5-step process, come out really super clean. And they come out in the latest version that we've been developing and that the dad in the lab is taking forward. They should come out aseptic, and sort of contaminant free, literally just pure sporozoites. If nothing else, that gives us an incredible tool to use in vaccine development, but it could also, it has the potential because you could have parallel machines running to actually manufacture something which is vaccine-able, if you like. Also in vaccine trials in mice, not in humans, we got equivalent control than we did with dissected. And we even started, just on the right, we even started to get some efficacy when you give an intramuscular injection of sporozoites. Now, the only way you can get protection at the moment with a whole sporozoite vaccine is to give it intravenous. That's not really going to work, I don't think, in the developing world. But intramuscular, we started to get protection. So that was, for us, really exciting, that maybe these parasites are so good, so pure, that they're actually even more immunogenic, which is we may get even more protection. So we can do potentially thousands, if not tens of thousands of mosquitoes per hour. We can now get them aseptic, they're hyperinfectious, they're as protective, maybe even more. And as I said, this could be a massive enabler for vaccinology, if not a vaccine itself. I do still want to ask the so what question, because I sort of feel like it's still a challenge. It's still a whole parasite vaccine. So if we really want to kind of address that challenge of cold chain storage and delivery in sub-Saharan Africa, this needs cryo conditions. Could we come up with the same kind of vaccine that didn't require -80 conditions, that could be delivered? Could we use some of the technology which is being developed today in RNA vaccines to deliver something which is similar? So just the last thing I want to touch on is, so my lab started to ask the question of, could we make an artificial parasite which had as many antigens as we could get, in particular to prime a T-cell response, but maybe even a B-cell response as well, and then put that into an RNA vaccine or an adenoviral vaccine that would deliver a huge cargo that would then launch a kind of an orchestra of an immune response, which would be protective like a whole parasite. So the first aspect of that was to find out what genes are on in the sporozoite. So this was a collaboration led by Eliana and Farah in my lab, together with Ginny and Mara Lornachak at the Sanger Institute, was to do an atlas of gene expression of the sporozoite itself. So we followed the sporozoite through its journey. It's really cool the way that, so this is blood stage malaria in red, gametocytes. You can see the males and the females separating out into 2 populations, the zygotes, then the sporozoites themselves. And as they go through their journey, they mature. And actually, when they are injected into human, they change their expression pattern again. So if we really want the best targets to go after, it may actually be important to go after targets which are in the activated, injected sporozoite and not those that are just sitting around in the mosquito. In fact, it could even be, and this is really, hand wavy, but it could be that the sporozoites you inject from a dissection or from a smoothie made mosquito are actually immature antigens, so the immune response might be suboptimal. So to now start to realize that there are proteins we should be targeting which are on the mature injected sporozoite, I think that's really exciting, and that's certainly an area that we want to explore. And so the last thing we're doing is, together with a small company spun out by Josh, the former postdoc in the lab, they've developed a machine learning algorithm that predicts T-cell epitopes, and that they stitch those together as a mosaic. And so we're basically working with their company, Base Immune, to develop a sort of an artificial parasite, a parasite that has multiple antigens in a string delivered by an RNA sort of plasmid or platform in a VLP or some other kind of delivery platform. And we're currently doing preclinical testing in a mouse model. And I'm really excited about that, because at least, we're going to give it a go. And if it doesn't work, then we'll fail happily. But I think the idea of at least trying to pursue something which is different, I think is really important in terms of the cargo that goes into the vaccine. So those are the 3 stories I've told you. This is the new team. which are sitting, for those of you online, you don't get to see their beautiful smiling faces in the front row. But we've just restarted the lab here in Sydney. The 5 on the screen now are kind of leading various projects which extend from the work I presented here. And then I guess the plan is that over the coming years is to really use those kind of insights to just constantly ask us the question of, we want to do really good quality foundational science, but we want to constantly have in mind this idea of bending the curve, of really trying to use that technology and that innovation to really start to do something meaningful to bring numbers down for malaria. I'll end with the acknowledgement slide. Both funders and people who've helped us will be involved with the work, and I'd be delighted to answer any questions. Thank you.
Thanks a lot. That was really fantastic. I have a few questions, but I'll give you in the audience the opportunity to go first, if anybody wants to start. If anyone, not, maybe I'll start then.
There's thousands of people here. They're all quiet. They're all quiet.
And I'll wait for people in the Q&A as well. I'm curious to know why you left out the blood stage vaccine candidates of your presentation, when you actually weren't, kind of found about RH5.
You can probably tell I'm somebody who kind of overthinks a little bit. And I went through this kind of period where I really sat down and I thought, if I'm going to go on for a vaccine, what's it going to be? A friend of mine was doing transmission, so I decided I'd leave transmission for them, because I think transmission blocking vaccines are a really good idea. And they do seem to have, in fact, the late Richard Carter, who died a year ago, he started to come to lab meetings, and he really influenced me in terms of transmission. That transmission is going to be a really powerful way to really start to address malaria control and eradication. So I'm a big fan of transmission blocking vaccines. I think the parasitic argument is if you get a good vaccine that works, you don't need a blood stage vaccine. So it should be good enough as a parasitic vaccine. We shouldn't need a blood stage vaccine. So that was kind of why I put my effort there. Why not? Why wouldn't we include a blood stage? I think there are candidates like the RH5 Ripper complex that you might want to include. I think my biggest fear of the blood stage vaccine as a concept is how on earth do you ever do the clinical trial? So you go into a community, you inject everybody with your experimental blood stage vaccine. At what point do you stop the experiment? I mean, people are probably going to get some level of blood stage malaria. Do you wait for 1% parasitemia? And then how do you know if you succeeded or not? So I think in my head, I've never quite got around the logistics of how do you prove that a blood stage vaccine works unless you use it in combination with something else. And if using it with combination with something else, I prefer to develop the something else first and then add on the blood stage as a sort of a fail safe in case anything breaks through. Oh, I mean, I might be wrong there. I don't know if others have thought about that similarly, but I've really struggled conceptually of how you'd ever actually bring a blood stage vaccine to market.
Okay, thanks. Any comments or questions?
Well, Mal should have a comment on that.
I'm just wondering what the struggle is with. I'm wondering what the struggle is with testing a blood stage vaccine. Wouldn't you just say, time to parasitemia above X, probably, time to thick film positive. And if that goes down, you've slowed the growth rate, presumably, of the parasites or reduced their density.
Yes, but have you put it? So I think in terms of the numbers game, I completely buy with blood stage vaccines. That if you slowed the rate, the impact would be massive. But for the individual, I'd just be worried that they might still die because you can have low parasitemia and still get all the immunological consequences of malaria. So I just the idea just started to make me nervous. Maybe I overthought it, but yeah.
Any other comments or questions from anybody else sitting here? And just reminding people online, you can, I think there's a button at the end, there's a button at the bottom. That's where you know that my English is, English is not my first language, but anyway, that's a joke about button and bottom, but there's a button at the bottom of the screen where you can.
It's not working.
No, I was just trying to give time to people to type the question on online.
I much prefer in-person questions, so you can always come to UNSW in person, knock on my door.
Okay.
Come and ask me.
I have another question then.
Okay, good.
Kind of, I guess, from the public health sort of perspective, and I mean, I guess that's related to the clinical trials or implementation of clinical trials, but kind of going back to that statement in terms of the statement that there is not a single malaria control intervention that can successfully lead us to elimination or eradication eventually. But do you think, and I'm just kind of, it's just, I'm teasing and just provoking you, like, but there are attempts of kind of upscaling the delivery of all the tools that we have.
Yeah.
And do you think there is that wouldn't be enough? I mean, the bed nets plus now, I guess, because I work on NTDs and we do MDAs and preventive treatments all the time, that with these, doing the same approaches that are being done now in settings where there is potential for elimination, where you go at it with the bed nets and also with the MDA. Do you think that would be enough?
Yeah. I mean, I guess the honest truth is I have no idea. I think my reading of the modeling literature is that that's going to work in some very well-defined. So the Gates Foundation and others called for this stratified approach, where if you know what's going on with malaria locally, you know what kind of intervention is needed. And I think some of the rollout of interventions is going to work in areas with maybe low transmission or very well-defined transmission, certain inoculation rates. I think, forgotten her name, from the London School of Hygiene, is it Meeks? She unfortunately passed away several years. Sylvia Meeks. Yeah. I met her once at a meeting and she said, if you want to eradicate malaria, start with Nigeria. Because if you can eradicate it there, you can eradicate it anywhere. That in rural Nigeria, it's so complicated, you're just never going to be able to roll out just conventional strategies. So in the back of my mind, I'm always thinking that I just don't, I'm very influenced by the modeling, but I just feel like we haven't quite got the toolkit yet that is going to work in the complicated areas. I think what we'll probably see in the next couple of years as they roll things out is the number of countries that are tackling the area get smaller and smaller. I think that is actually happening. I think maybe that's underreported, that more countries are declaring elimination. So I think it's going to be concentrated effects in these really complicated, often very politically unstable or war, famine, whatever, all that kind of jazz. Those are going to be the really difficult countries, and that's where something which is deployable like a vaccine is a game changer in my mind, because then you can have these campaigns like they're trying to do for polio. I mean, polio should be easy, right? We were kind of like waiting for the day when they say polio is gone, but it's still not quite gone. And I wonder whether that's what's malaria, the end game, is going to be like too. There's going to be areas which are really hard. And I think only something, in my mind, something like a vaccine, diagnosis and vaccine is going to be really, really important. But yeah, I'm not just trying to keep myself in employment. I'm very happy to be proven wrong.
Yeah. And yeah, thank you. Anyway, I mean, I guess the technology that you're developing is really, or trying to develop, is really translatable to other diseases, that even if malaria is gone, you would have other diseases to focus on. Any questions or comments? Tony.
Jake, that was a great talk. It was really, really excellent. The drug that you're going to treat mosquitoes with, can you tell us a bit more about that? I mean, is it difficult to synthesize, and have you got any idea of its stability and that sort of thing on sort of nylon or plastic or whatever a bed net is? Or
I mean, this was literally the last project we did in the lab before I moved to Australia. So we can synthesize it, so you can make it, and you can actually derivatize it, which is why this drug was so attractive in terms of the project, because we could do things like we could add a click handle on it, we could make a photoactivatable handle on it at the other end, so we could use photoactivation to click it to its target. So that was kind of really cool to be able to do that. And that means that there's a whole, and we've already done one paper on medicinal chemistry of the drug, and I think then that's the problem, is we're not really a drug development lab. You really need a strong chemistry lab that can do the kind of the rigmarole of stability and things like that. So the quick answer is I don't know how stable it is in the environment, but it's definitely a scaffold that has enough ability to be modified that I think you could do quite a lot with it. And it still may not work, but I think for me the concept kind of was a light bulb moment that actually some of our efforts in drug discovery don't need to go into humans. And that's Flaminia's whole kind of charge.
Well, as you observed, the pathway to regulatory approval would be far cheaper, right?
Yeah, absolutely. And also, it's not just bed nets. I mean,
no, exactly.
It's like sugar baits in the environment, and it doesn't even have to be toxic. It can just be. I mean, it's like giving a tiny little pill to a mosquito. It's like a really cool idea. So I think there's a lot of mileage there if we, just thinking laterally. I think that's where solutions may come from, like really lateral kind of ideas.
Thank you. Okay, one last chance for people online. I don't see any questions there. Anybody else here? If not, once again, thank you so much.
Pleasure.
I'll be knocking on your door.